Today we're exploring a topic that's gaining serious attention in the scientific community: whether a simple and widely available amino acid, arginine, might play a role in slowing one of the most destructive processes associated with Alzheimer's disease. This doesn't mean we're talking about a cure or a proven treatment, but recent preclinical research has revealed findings interesting enough to deserve a deeper conversation. In this video, we'll look at what these studies actually show, why researchers are cautiously optimistic, and what all of this might mean for the future of Alzheimer's research, without hype and without promises. Alzheimer's remains one of the most challenging neurodegenerative conditions to treat, affecting tens of millions of people worldwide. One of the reasons it's so difficult is the accumulation of amyloid plaques, sticky clusters of beta-amyloid proteins that build up in the brain and interfere with neural communication while triggering inflammation and progressive cognitive decline. For decades, researchers have tried to stop or slow this process, but most attempts using traditional pharmaceuticals have shown limited success. This is why scientists have begun exploring a new angle: the possibility that modulating basic cellular processes, including the availability of certain amino acids, could influence how these plaques form. Arginine became part of this conversation because of several studies showing that low levels of this amino acid are associated with increased plaque accumulation in animal models. Arginine is already known to play important roles in immune function, blood flow, nitric oxide production, and cellular repair. When researchers noticed that animals with lower levels of arginine tended to accumulate more beta-amyloid, they asked a simple question: could restoring or increasing arginine levels help slow down plaque formation? A research team in Osaka, Japan, recently decided to investigate this question in more depth. The new study, conducted at Kindai University and published in the journal Neurochemistry International, examined how arginine interacts with amyloid beta, both in the lab and in animal models. In the initial laboratory experiments, scientists observed that arginine behaved like a chemical chaperone, meaning it interfered with the ability of amyloid beta proteins, especially the highly problematic AV42 form, to clump together. The more arginine present, the slower these protein aggregates formed. This alone was promising enough to justify moving forward with animal testing. When the research team administered arginine orally to two well-established Alzheimer's models, fruit flies genetically engineered to express amyloid and mice carrying several Alzheimer's-related mutations, they found that the animals given arginine developed fewer amyloid plaques, had lower levels of insoluble AV42, showed better behavioral and cognitive performance, and displayed reduced inflammation, including decreased levels of pro-inflammatory cytokines. Taken together, these findings suggest that arginine might interfere with plaque formation, support the brain's ability to clear toxic proteins, and help calm the inflammatory responses that accelerate neuronal damage. This combination of effects explains why scientists are paying close attention. However, despite how exciting these results may sound, it's important to be clear: we do not know if any of this translates to humans. The researchers themselves emphasized several key limitations. The doses used in animals do not correspond to typical supplement doses in humans. The findings so far are limited entirely to lab models and animal testing, and there are currently no clinical studies testing whether arginine has any meaningful effect in people with Alzheimer's disease. Because of this, arginine should not be used as an Alzheimer's treatment or for prevention. We simply don't have the evidence. Even with these limitations, scientists are interested in arginine because it has a strong safety profile, is inexpensive, is already used in clinical settings, and could, if future trials prove effective, become a low-cost and widely accessible component of a broader therapeutic strategy. This makes it a potential candidate for drug repurposing, a process in which existing compounds with known safety records are evaluated for new therapeutic applications. But until clinical trials are conducted, everything remains theoretical. What this research does highlight, however, is how deeply the brain's biochemistry is influenced by core nutrients and metabolic pathways. It suggests that future Alzheimer's research may involve a combination of metabolic, nutritional, and biochemical interventions rather than relying solely on a single pharmaceutical breakthrough. The idea is that multiple small changes across different systems could create a meaningful overall impact. For now, arginine remains an intriguing scientific lead, but not a proven therapy. If future human trials confirm even a portion of what has been observed in animals, it could open the door to new approaches for supporting cognitive health that are accessible and affordable. But until that evidence arrives, the responsible approach is to view these findings as promising but preliminary. If you want to stay updated on breakthroughs in brain health, nutritional neuroscience, and metabolic wellness, make sure you subscribe to the Vital Nourish. And now, it's your turn. What do you think about nutrient-based approaches to brain health? Do you believe metabolic therapies will become a bigger part of future treatments? Share your thoughts in the comments. Thanks for watching, and remember, caring for your brain is one of the most valuable investments you can make.